So our speaker today is Zhong-Hua Chen. Zhong-Hua did his undergraduate and master's degrees in China at Zhejiang University, and please accept my apologies for my pronunciation. And then he did his he did his PhD at the University of Tasmania in Australia. He's also done a postdoc at Glasgow University in the UK, so he is very well travelled. He's been now at the University of Western Sydney in Australia for a significant period of time. Obviously, he's experiencing a few personal challenges being in Sydney within a lockdown at the moment, but nevertheless, it's a great opportunity for him to present his work here today on potassium transporters in barley. So we're very much looking forward to your presentation, Zhong-Hua. Please, if you're in the audience and you've got some questions, put them in the chat. Zhong-Hua will speak for about 30-35 minutes, and then we'll take some questions towards the end of the presentation. So please put those questions in the chat. I will do my best to translate them, and I hope you enjoy the session. So Zhong-Hua, great to see you, and hope it's an exciting and interesting seminar.
Thank you, Ian. Thanks, Elma, for organizing this seminar. Today, I'm going to just give you a talk about our recent work on potassium transporters. So part of this work has been published on Journal of Experimental Botany last year. So this is mainly work from a group of people. Initially, I recommended Dr. Xue Feng, who's the first author of the papers, but she's having a baby. So here I'm representing the group to present this talk. First, to just give a bit of background, drought is a very serious issue across the world. So in almost all the continents except Antarctica, drought happens very often, and especially in Asia, Africa, and also Australia. Despite Australia is a small continent, drought is a big issue. Frequent drought events in Australia, in all states and territory. So here are just a few images showing the drought effects. In Australia, drought has been accompanied Australian agriculture since early settlement, and we recently had a big drought from 2017 to 2018. So drought affect the agricultural productivities in Australia. You see affecting the grain industry. So it also affect barley, which is one of the main exporting crop in Australia. That's why we study barley. Long-lasting drought reduced food production in Australia. So in the early days, in 1877, all states affected by severe drought, especially in Queensland, which is a state in the north east of Australia, and also in Western Australia, a lot of crops failed. Not long after that, in 1902, a lot of land and sheep are affected. So effectively, the production of animal and crops are severely affected. During the Second World War, Eastern Australia again suffered a drought condition, lasted for almost 10 years. So those long-lasting drought has really impacted Australia. Although Australian farmers are very resilient, long drought really devastating for their livelihood and also for the productivity. 2003, very long, probably the longest in record of Australian agriculture. So that affected Australian productivity of the crops. In recent years, not long ago, just before the COVID, we had 3-year below average rainfall and drought across many states. And this also caused a very widespread of bushfires. So that affected not only agriculture, and also affected people who live in the city. So I remember in 2019, the Sydney Basin, where Sydney the city is, we're surrounded 3 sides by bushfires. The only place we can get out is the sea, but the smoke was so bad, and that's mainly due to low rainfall and drought condition. Similar situation, drought in China. China is a big country. There's many different provinces, and in the major wheat and barley producing provinces like Xinjiang, Gansu, Henan, Shanxi, Shandong, those provinces, drought are very frequent. And those northern and northwest provinces usually already have rainfall, so they are inland. Drought will really lead to large crop loss. Here is just a table summarizing the drought effect on crops. So it can cause around 50% of yield reduction if drought hits at seed filling stage. And barley is a relatively tolerant crop species to drought. So around 400 millimeter of rain should be enough to support barley. But drought is condition that the rain could be very low. So that's still barley still cannot tolerate extreme drought. So our focus is on potassium transport. Potassium is one of the macronutrients, the 3 major ones, NPK, one of them. Potassium is very important to maintain membrane potential in plant cells, and potassium is also a major nutrient or major ion for stomata movement or regulation. Potassium has a major role in root elongation. So across different tissues in the plant, there are many types of potassium channels, co-transporters, and antiporters. So, for example, in the roots, there are a range of the shaker type K channels, AKT1, 2, KC, GOG, and also the other co-transporters. In the leaf, different type of K channels and transporters, and also stomata and other parts, even in the reproductive tissue. For example, in the pollen, SIPK is a potassium channel for pollen germination. So potassium channel, quite a few large families and many family members among many different plant species. They have a very important role in plant growth and stress tolerance. So our focus will be on AKT1, AKT2, and HvAKT, 3 ion channels, and the functional analysis are only limited to the barley genes. This is just a simplified tissue-specific potassium transport. So in a different tissue, in root, xylem, mesophyll, phloem, there are many different types of potassium channels. You can see the AKT1 and HvAKT, AKT2, those are functioning at different level. So for the barley HvAKT1, the focus was on root because it mainly expressed in roots. For the AKT2 and HvAKT1, the focus of our study was on leaf. just to clarify this. So before we go through the molecular cloning and functional analysis, so we first did a range of the molecular evolutionary analysis. So we use blood method and the hammer method to do protein sequence similarity analysis. We found those many of those potassium transporters, including the KAT or AKT, they are the same shaker type channels, may have evolved from algae, either streptophytes algae or chlorophytes algae. Those are large groups of algae. And those green algae are the assisted to the ancestor of the current land plants. For HvAKT1, we first conducted a phylogenetic analysis of a putative HvAKT1 protein. So those are the ones we identified in the 1kp, 1,000 transcriptome, plant transcriptome database. So this was published in 2019, and there are over 1,300 plant and algae species. So we found a lot of them in HvAKT1 in those species, and we select the important ones representing angiosperm, genome sperm, fern, lycophytes, moss, liverworts. So all of those are land plants, and streptophytes, chlorophytes, and those are the green algae, rhodophytes, red algae, and Chromista is another algae species. Algae group. We use this as an outgroup for constructing trees. Protein sequence alignment and structural analysis show that it's highly likely, especially the K-selective filter, the very conserved GYGD residue you can clearly see it's in barley, it's in pine tree, in fern, and also in the streptophytes algae. But this was not identified in a chlorophyte algae. So we propose that potentially those the HvAKT1 and its HvAKT1 is originated from streptophytes algae. Then we analyzed HvAKT2, similar story. HvAKT2 for highly is another potassium channel, have different function. HvAKT1, based on the study in Arabidopsis and the rice, HvAKT1 requires CBL and the CIPK protein kinases to activate, and HvAKT2 itself forms a very weakly voltage-dependent ion channel. And HAK is a K transporter, co-transporter, but the typical component of K-selective filter is also identified. You can see this GYGD is available across the major line ages of plant species, and also exist in the basal range of streptophytes algae. Here is the Clebuso flaucidum. So this is one of the basal species, and one of the species have a genome sequence. Recently, we did some work to identify or propose some of those typical shake type K-channel structure and their potential interacting molecules. So here is just a model showing the 6 transmembrane span domains of the shake type K-channel that can be outward rectified, which would be HvAKT1, HvAKT2, or KAT1, KAT2, or a outward rectified potassium channel that the representatives are GOK channel and SCO channels. So there are potential interacting molecules like SAMP, CGMP, GABA, GBRO, protein, ATP. So this gives us a bit more insight into the structure. Of course, there are a lot more study done in Arabidopsis, so I won't go into too much detail because this talk is focused more on barley. Stress-induced ROS-mediated homeostasis. So stress under many of the stresses in plants, biotic stress or abiotic stress, reactive oxygen species plays a very important role. Sorry. Can you turn off the? Okay, that's fine. All right. We found previously potassium efflux channel linked and activated by ROS, and that is linked to stress-induced programmed cell death. So we also measure ROS in our study, and same for nitric oxide. Nitric oxide is very important, a small molecule in plants, and they're linked to stress tolerance in plants. Again, we did genetic similarity analysis. So those are the key gene families. And here you can see ROS and NO signalings. Those are highly conserved across many species. So you can see across angiosperms, genome sperm, fern, lycophytes, moss, all the way to streptophytes, algae. And some of them are even more conserved. You can identify them in even more ancient chlorophyte algae. So all those are in barley as well. As you can see, barley is here. So with all those basic bioinformatics and molecular evolution analysis, now we get into the functional analysis of those potassium transporters. We cloned all those 3 genes, and we found barley AKT1 is localized in the plasma membrane, and it's, of course, as I mentioned earlier, it's highly expressed in the root, and it's also induced by drought. So we use 3 different barley variety to see there are different levels of expression. So we used both onion epidermal peel and also barley mesophyll protoplasts to express the gene and to localize. So we can clearly see the G35S HvAKT1 is on the plasma membrane, and the IFP is a plasma membrane marker gene. So when you have the overlay, they merge together. So clearly showing that it is a plasma membrane localized protein. And we use a transient silencing technique, Figus technique to silence the HvAKT1, and so we clearly see when after silencing the HvAKT1 in barley, the drought tolerance has reduced. You can see from the visual, the picture. And here we didn't really use actual drought. We use PEG, polyethylene glycol, as an osmotic stress or mimicking drought stress. And the potassium concentration has significantly reduced when the HvAKT1 is silenced, and the potassium concentration is also reduced. So overexpression. So we generated some stable overexpressing lines. So here we only show one line, but we have generated a few different lines. Overexpressing HvAKT1 as slightly significant improved drought tolerance of barley, but I won't see it as like what we would expect a much bigger, much better improvement of drought tolerance. But the reality is the improvement isn't that great, but still statistically significant. But you can see the biomass overexpressing HvAKT1 significantly increased the biomass under drought condition. And then we did some electrophysiology measurements to look at the response, ion flux responses. So we use a equipment called micro electrodes ion flux measurement, MIFE technology. We tested AKT1 either by silencing the AKT1 or overexpressing AKT1. You can clearly see we use a PEG here as mimicking drought. So when you silence the HvAKT1, when you add PEG, and usually the control plants showing a sign of taking up potassium, which you can see a positive net potassium flux. This is quite commonly observed in not only in barley and also in other crops. So PEG stress will increase potassium uptake, at least at transient at the first 20 minutes. But this silenced BSM V silenced plant does not show any significant potassium influx, and the flux is much lower if we measure that over the 24-hour period. For the overexpressing line, potassium flux has been slightly improved, although the golden promise, which is the wild type, also shows some pretty significant potassium influx under PEG treatment. Interestingly, we also found that as you regulate or genetically modify a potassium channel in barley, there's also a very obvious significant response of proton homeostasis. So we can clearly see using electrophysiology, there is a significant difference between the the pump activity. So the sign positive means efflux. So here you can see the when you silent the AKT1 actually affecting the proton pump activity as well. The control plants showing the negative ion flux. That means the pumping, the proton pump is pumping proton out in order to take up nutrients like potassium, calcium, and other nutrients. And also the proton ATP's activities has also declined in the silenced line, but this is increased in the overexpressed line, the expressing line. So quite interesting finding for modifying a potassium channel, also regulate the proton pump. For the secondary messenger like nitric oxide and hydrogen peroxide signaling, we can clearly see that when when the plants silenced with HvAKT1, there is a decline of nitric oxide production under drought, but an increase of hydrogen peroxide. So this is again quite interesting findings of the paper. And this that was total content, and we also measured in the root using a fluorescence dye to test the nitric oxide and hydrogen peroxide in the root. So the fluorescence signal shows the similar trends as compared to the total tissue nitric oxide and hydrogen peroxide content. Okay, next we move to the other two genes. The reason we put those two together are because there is we were not able to identify the interaction partner for HvAKT1. So we actually did a fair bit of work on using yeast to hybrids, and also we did oocytes work, but we could not identify the similar similar CBL1, CBL9, and CIPK23 as in Arabidopsis. Those are the two combined what we call protein kinase combination activates AKT1 channel activity. Without those two, AKT1 is silent. But here we found that AKT2 and HvAKT1 both have a current without kinase activation. So we first tested the relative expression of the two genes. AKT2 is obviously highly expressed in leaf as compared to root and stem, and HvAKT1 high affinity potassium channel are expressed in highly in both leaf and root, slightly high in leaf. And we use in situ PCR to localize to locate the two genes. You can see they are in the bundle sheath, the blue stain, and also in the root. So no RT, that's no real reverse transcription. So that serves as a negative control. And we also use HvAKT for running the in situ PCR to use as a positive control. So clearly, those two genes can be identified in both root and leaf, but they are more abundant in the vascular system. So that's for potassium transport. So it's quite clear for the tissue expression. And then we also cloned those two genes and identified them, and they are both plasma membrane localized, although the signal isn't that very strong. Even we use 35S, quite strong promoter. I think the reason behind is because usually on plant cells, those ion channels, like the ion channels, are quite big protein, and they too many of those their expressions are usually quite low. Too many of those ion channels on a plasma membrane will cause issues for plants. They are not highly expressed, but despite that, we can clearly see that overlaps. The GFP overlaps with the plasma membrane marker. We also used the Xenopus lavis oocyte system, then 2 electrodes voltage clamp to test. So effectively, we express, we've synthesized the capped RNA of those two genes and express them, inject them into oocytes. Then measure the use electrophysiological method to measure the current. Here is just showing the selectivity. So both ion channel and transporters are selective to potassium over other ions, over sodium, cesium, lithium, trace, or rubidium. So rubidium has some conductivity, and HvAKT2 actually is a very weakly voltage dependent, and the current isn't very high. So silencing the two genes well, separately confers Drought tolerance in barley leaves, and clearly see the decrease of the decrease in the silencing lines, and also slightly and this is very clear, and also the K concentration dropped. So that means the channel the function has been disrupted, well, not fully, although it's not fully suppressed. Overexpressing of HvAKT2 and HvAK confers Drought tolerance in barley leaves. So in leaves, again, here we're only showing, just for the clarity, we're only showing one line, but we have multiple lines data. So those two genes you can see in the overexpressing line, few times higher than the control, and that increase significantly the biomass under drought conditions. However, the increase isn't that great as what we expected. So I'll talk about that at the conclusion. So phenotype is quite obvious, and it worked to some extent. So overexpression of those genes will improve drought tolerance, especially in the leaves. And again, here we found that regulating those two ion channels also affecting the proton transport at gene expression level and enzyme level, and also at the physiological level. So this figure shows that HvAKT1, so that's a proton pump, the proton pump gene, is significantly reduced. The expression is reduced in the two silenced lines, but this the expression is increased, although it's not like a few fold difference, but there are some difference. And this is also evident by the enzyme activity assay, similar trends. So the proton flux, as I mentioned earlier, a negative proton if the proton efflux is usually a good sign of good proton pump activity, and in the control, there's always negative pump negative flux. However, the silencing of those two genes lead to a positive or net zero flux. So the pump activities are significantly affected in those silenced line. On the contrary, that when overexpress HvAKT2 and HvAKT1, the proton pump activity has been restored, and in some cases those activities actually increased. So this is kind of to our surprise, but that's what we observed. So quite interesting results, and those are measurements from barley leaf. So measure mesophyll ion flux from the mesophyll cells, not isolated mesophyll protoplasts. We actually peel the leaf and sit there for some time to stabilize the leaf, then measure ion flux from those mesophyll cells. Here are some evidence showing the net flux changes of those transgenic lines. Potassium flux is higher in the two overexpressing line, but it appears that overexpressing HvAKT2 has a slightly better recovery of the uptake in response to PEG treatment. The silencing those genes would cause a reduction, a very significant reduction of the potassium flux, and the potassium flux are close to zero. So there is really no net, very small net potassium uptake. Proton flux, I mentioned in the previous slide, there isn't really much trends for the calcium flux, although calcium is quite important. We can clearly see there is actually an increase of calcium influx in the BSM-V HvAKT2, but the rest are not really significant. So this could be a sign of a calcium signaling over the time of 1 to 12 hours, but that returned back to a level that isn't significant between all those lines as compared to the control. Okay, the last few slides. HvAKT2 and HvAKT1 respond to drought stress also via the regulation of nitric oxide and hydrogen peroxide. Again, similar results as we seen for the HvAKT1. We apply SNP, which is a donor for nitric oxide, and DMTO, which is a hydrogen peroxide scavenging. So we see the phenotype, and CPTIO is a nitric oxide scavenger or inhibitor. Okay, I think of time. Okay, to summarize, here we found plant AKTs, HvAKT1 and 2 and HvAKT1 may have evolved from streptophytes algae, but at what exact species and where are still not very clear. So we are doing some further studies, and we're also trying to clone some of those AKTs and HvAK from algae such as Klebs and Chara. Chara is one of the streptophytes algae or charophytes recently been sequenced and with the full genome. Silencing those three potassium transporters result in significantly decreased biomass, suppress the potassium uptake and reduce the potassium influx under drought treatment in barley. Overexpressing of those three potassium transporters channel improves the drought tolerance. So those are mainly linked, especially with proton homeostasis, and of course disrupt those potassium transporters will definitely reduce, will affect the potassium uptake as well. And also there is a significant role for nitric oxide and hydrogen peroxide production in root and leaf cells, regulating drought tolerance in those transgenic lines. Our results highlight the importance of potassium transporter and potassium nutrition in response to drought stress. However, there's a limitation. As I mentioned earlier in some of the slides, those transgenic barley plants do not achieve high tolerance as what we expected. They are good. We can see some improvement, but they are not good enough for field trials. And also, this is I think it's reasonable because disrupting or overexpressing or silencing one of those transporters can only partially affect drought tolerance. That's my conclusion, and you can understand that it's because, as I mentioned earlier, there are so many potassium channels and transporters around and controlling. So, doing transgenic work on one single gene may not be very effective for drought stress, which is a very complicated traits in crops. And so far, I've never seen any group claims a transgenic plant with one single gene that can actually be super tolerant to drought. Okay, so I would like to acknowledge a few funding bodies in China and also in Australia. I also would like to thank the Society for Experimental Biology for the opportunity to present our research work. I still remember I attended the SEB meetings in 2009 when I was in the UK. It's a very nice meeting, and yeah, and I really enjoy the experience. And also, I would like to thank Journal of Experimental Botany for publishing our research work. Really appreciate the support, and thanks for your time to listen to my talk. So I'm open for some questions.
Great, thank you, Zhong-Hua. Real tour de force of electrophysiology there. Huge amount of data that you and your group have generated recently. So congratulations, obviously, on the paper in Journal of Experimental Botany, and also, you know, for presenting today. So I've just been monitoring the chat, and it's very quiet on the chat. I was hoping to see a few questions in there, so I might start with a question of my own, and that is, you show these differences in biomass when the transporters are overexpressed or silenced. Looking at the plants that you showed in the images, they seem to tiller fairly normally compared to the untransformed plants. And I'm just wondering whether there was changes in photosynthesis or leaf elongation that underpins the differences in biomass that you see.
Yeah, we do have the data for photosynthesis, and the actual difference between the wild type and the transgenic lines are not great. So although they we don't observe a very obvious phenotype, but that's relatively. So the plants, I think, are slightly smaller. The silenced plants are smaller, and the overexpressed plants I would say slightly bigger, but not significant. So just observation.
Yeah, and with those measurements, Zhong-Hua, those were sort of typical infrared gas analysis measurements at a particular point in time, were they?
Yes, yeah, we didn't because we mainly use at seedling stage and also, of course, at reproductive stage. But we only did kind of spot measurement. We didn't do like weekly measurement over a long time. So I think this is actually a very good suggestion. We still have the seeds, so we can probably do some more detailed physiological measurements of those transgenic line. As we're not planning to do field trials because they're not they're not great lines with a big agricultural potential, although this transgenic, so it's probably not popular anyway. So
sure. Marika has asked whether there was any movements, any measurements of stomatal movement kinetics in any of the lines that you've generated. And I think you've already answered that question, but I know there's a lot of information emanating from the likes of Steve Long's groups and other groups that are looking at manipulating photosynthesis, where there's been a lot of attention given to the dynamics of stomatal movement, and particularly how photosynthesis and stomatal conductance responds to change in light environment. I wonder whether this more refined phenotyping approach might give some clues as to why you've got this higher biomass in the plants where the potassium transporters have been overexpressed.
Yeah, I think that's a really good suggestion, and I think I myself work a lot on stomata, so we didn't really pay too much attention because we didn't see a very strong stomata phenotype. We did also a transgenic work of a few other genes on barley, and we found a couple of them are actually having stomata phenotype, but this one we didn't pay particular attention. But I think this is something, yeah, I'll take this message back and talk to my students and the previous PhD student. They're now like working in other universities. They got their degrees, but it's yeah, I think it's very useful for us.
Yeah, I think these sort of
are very useful. Yeah.
You know, these punctual measurements of leaf gas exchange that, you know, most of us don't always pick up, you know, the sort of subtle phenotypes that you might expect when you manipulate a single transporter where there is obviously considerable redundancy of function in the plant.
Yeah.
And I think some of these more subtle approaches, like looking at how gas exchange responds to changes in the environment, such as light, such as vapor pressure deficit, can be important. And one thing that my own group has been looking at recently, we had a very talented postdoc who built a sort of whole plant gas exchange chamber where we can control the vapor pressure deficit around the whole plant. And so we're using this as a sort of screening tool to see whether there is genetic variation in whole plant response of both.
Well, that's great. Yeah, that's good. I would like to do some collaboration in the future if it's possible, because I think the whole plant chambers, as far as I know, they're only available for Arabidopsis. So you guys built like house-made, like custom-made chambers?
That's right. Yeah, it's a custom-made chamber. We built it for some work that was supported by the International Wheat Yield Consortium. This is a collaboration.
So wheat.
Yeah, so we built it for wheat, but we've also grown broad bean, dwarf tomato plants, a whole range of species in it, and it does provide very nice control of vapor pressure deficit around the leaves. And you know, we've certainly used it to screen for genetic variation in the response to vapor pressure deficit, but you know, we're also using it to look at how the whole plant responds to stresses such as soil drying. So, you know, if you're in the UK when lockdown finally ends, you know, you're more than welcome to come and use it, Zhong-Hua. Join the queue.
Yeah, that's great, because my new ARC project will be I'll be working on wheat and barley stomata regulation. So if we can use your powerful whole plant chamber to measure those, I think that will generate good data, because we identified some genes and we've generated some lines, but it would be great to have those tested at whole plant level.
I'll send you
I
the.
plan to visit UK. Yeah, yeah. If you can send me some, I'll send you an email after that, and we
Sure.
on collaboration.
I'll
Yeah.
send you the reference. I mean, the other point that I sort of had as a question, I think you showed a nice data set where there was quite considerable genotypic differences amongst a range of barley cultivars in the expression levels of some of the transporters. And I was just wondering whether if you have, I mean, clearly if you silence or overexpress a particular transporter within a genotype, you get these changes in drought sensitivity and drought tolerance. And I was wondering, across a whole range of barley genotypes, if you can look at the expression levels of the transporters, whether that actually gives a sort of screen for drought tolerance across a range of genotypes.
Yes, we actually did a fair bit of work, and which I didn't present here. So we use a lot of wild barley and some landrace and of course elite cultivars. Yeah, some of those genes, the expression are quite different. So they're like at normal condition, and also we compare them at drought treated. So yeah, some of those work are done, but not so systematically. So one student came in and did some work, see some difference using 10, 20 lines. Yeah.
Does anyone else have any questions at this point? Because otherwise I'm conscious that Zhong-Hua and I would probably continue discussing lots of aspects of barley drought tolerance for another few hours, I suspect.
Yeah, so the major stomata movement kinetics in any of the mutant, especially in response to ABA, yeah, we did some measurement, but not really using epidermal peels or response to ABA. So this is something we will take those comments back and do some measurements later on.
But perhaps if I could ask just one final question, because I realize we've been on the call for close to an hour now. I mean, I'm interested that you've investigated drought tolerance specifically, but I would expect there'd be considerable interest in looking at salinity tolerance of these lines as well. And I was wondering whether your group has started to investigate salt tolerance of some of these lines as well.
Yes, this is in our current work. So yeah, of course, I also work on salinity. So that's kind of I only start work on drought since from the time when I joined Mike Blatt's lab. So I've been working on salinity longer since my master's time. So salinity then PhD. So yeah, obviously that's something we are also looking and interested to do something in the near future.
Well, clearly you have a range of genetic resources available now with these silence and overexpressed lines. I mean, personally, I find barley a very nice plant to work with, particularly because its leaves are large enough to clamp a pyrometer on if you're interested in stomatal measurements. I've spent a lot of time in my PhD actually measuring elongation rates of barley leaves in response to different stresses. And it's a very nice plant to work on, I have to admit, notwithstanding its
I think barley was the genetic model for cereals before rice and Arabidopsis took over. So my PhD was on barley too.
So you've maintained the link.
Yeah, I think so. Yeah, I think I published. Probably more than 50% of my papers are published on barley. So
a true crop specialist. Then I can't see any further questions coming in, Zhong-Hua, in the chat. I don't know whether Elmer, you wanted to say anything by way of announcement for the next Journal of Experimental Botany seminar, or Marika.
Yes, most JXB webinars will be shown on our SCB website on our events page. So you can check the next events there.
Okay, thank you very much, Elmer.